For this experiment, we asked ourselves, how do stress factors affect daily cortisol levels in captive common bottlenose dolphins? Dolphins in a captive setting can be occasionally subjected to a variety of potentially stressful situations. The stress response comprises a variety of biological reactions to internal or external stimuli elicited when an individual perceives a threat to its homeostasis, according to Moberg and Bench. Noise can act as a stressor to dolphins because they rely heavily on sound for many important life functions, so it can be inferred that exposure to noise may have detrimental effects to important life functions. Sometimes physical movements cannot indicate that a dolphin is stressed, so we tested stress levels by reading cortisol levels through the saliva of these bottlenose dolphins. Cortisol is released through the endocrine system in the hypothalamus as a result from stressors to homeostasis.
Repeated daily collection of dolphin saliva can help to understand how acoustic disturbances can provoke changes in dolphin homeostasis. The objective of our study was to assess the acute stress response to anthropogenic stressors in a group of common bottlenose dolphins. Salivary cortisol levels were analyzed by enzyme immunoassay on a day with construction noise and were compared with those from 3 regular days at the aquarium with no construction. The study was conducted on 5 adult bottlenose dolphins. The group consisted of a breeding pair, 2 subordinate younger males, and a subordinate younger female. The 5 dolphins are trained for voluntary saliva sampling. For saliva collection, the dolphins slid out of the water onto the training platform and emptied the mouth of water by tilting the open mouth downwards. Saliva samples were collected with absorbent swabs placed laterally to the base of the tongue for 5 to 10 seconds. The swab was placed in a tube, centrifuged, and the saliva obtained was stored at -20 degrees Celsius until analyzed. For each animal, samples were obtained 4 times per day for 3 regular days, as well as for the 1 day the facility underwent construction. The construction consisted of drilling of the walls surrounding the different pools at regular intervals over a 10-hour time frame, starting at 8 a.m. and finishing at 6 p.m.
Please refer to the figure on the screen as I discuss the results of the experiment. During the control days, average salivary cortisol levels at 8, 12, 15, and 18 were 0.13, 0.19, 0.1, and 0.14 nanograms of cortisol per milliliter of saliva, respectively, whereas the day of construction works, hormone levels were 0.1, 0.27, 0.86, and 0.35 nanograms of cortisol per milliliter of saliva. Figure 1 shows the average salivary cortisol levels of the standard days at 4 time points. These results follow the same pattern previously described in plasma cortisol of Indo-Pacific bottlenose dolphins, where serum cortisol levels declined in the evening and increased the next morning. During the day when construction works were carried out, all 5 dolphins showed increased salivary cortisol concentrations with a p-value of 0.0004. This was more evident in the breeding male, who showed a 300-fold increase of cortisol level in the afternoon. The assay detected an acute increase in salivary cortisol induced by vibrations and noise in the dolphinarium.
Through our research findings, salivary cortisol measurements that were obtained 4 different times during a whole day displayed variations similar to that previously established for plasma cortisol. Construction around the dolphin enclosure took place during the day when the cortisol level spiked. Therefore, we can conclude that vibrations and noise in the dolphin facility from construction work induced an acute increase in the salivary cortisol. However, when compared to wild bottlenose dolphins, the circadian rhythms of salivary cortisol were similar. This factor makes it difficult for our study to differentiate between the natural fluctuations of stress and those induced by the constriction and vibrations. We were also not able to obtain results from a large number of bottlenose dolphins to better clarify our results. The study raised the questions of how can stress levels be lowered for bottlenose dolphins in captivity, and what other studies can be performed better to understand the stress levels of captive bottlenose dolphins. In view of these results, a project for ascertaining salivary cortisol baselines and its specific variability by means of multi-facility simultaneous investigation with a large number of dolphins is being developed throughout Europe under the guidance by our co-author, Javier Almunia.
Further research is in the works to observe the effect of noise on cortisol levels, but in larger sample sizes of dolphins. We need to collect data from more dolphins to get a greater idea of how exactly the noise is affecting them. We could test types of noise and duration of noise to see if they affect the dolphins differently. After we have found out these things, we can report them to aquariums and dolphin sanctuaries to tell them how they can best keep their dolphins happy. Further research should be also conducted on methods to lower salivary cortisol levels in captive bottlenose dolphins and marine life in general. We believe that cognitive enrichment may be one way to help lower their cortisol levels. Cognitive enrichment is gaining popularity as a tool to enhance captive animal well-being, but research on captive marine animals, such as dolphins and whales, is lacking. Dolphins are very intelligent animals that need a creative outlet while living in captivity. In most cases, the enrichment of choice is to put out a few balls in their tank and hope the dolphins will be entertained by it. This will quickly lose the attention of inquisitive dolphins. In future experiments, we could try underwater mazes for the dolphins to navigate through with some type of reward awaiting them at the end. Similarly, some type of puzzle for the dolphins to solve daily may provide some stress-free enrichment, maybe hiding treats for them. Allowing the dolphins to hunt their own live fish should also be considered, as it is a time in the wild for the dolphins to communicate with one another and work together to capture their prey. Another example for their enrichment. Human interaction with the dolphins may also prove beneficial, even if they are just bringing in a toy for the dolphins to retrieve and bring back to them. Dolphins are very active and enjoy interactions with others. We could then test the dolphin saliva again to see if their cortisol levels decrease, increase, or remain the same with the addition of these various enrichment activities. Perhaps another way to gauge the effectiveness of this enrichment activity could be to see if the dolphins exhibit less of the stereotypical tendencies, such as swimming laps around their tank, excessive whistling, and/or biting the gates that they live in, that come along with boredom of dolphins in captivity.